site-specific recombination, possibly the enzyme system that Jay was using in his experiments, are a valid alternative to DNA ligase for quantifying loop formation in DNA. And then I'm going to switch for the second half of the talk to some computational work that Stefan has done. And the objective here has been to develop a general and direct method for calculating topological free energies for any arbitrary DNA topology. And by this I mean for DNA knots, links, supercoiled molecules, as well as DNA loops. I hope also, this is unpublished work, Masa's work was published very recently in NAR, but I hope I will be able to convince you that we've actually succeeded in this challenging task. So I'm going to spend a little bit of time reviewing, probably I don't need to do that for this audience in detail, but just going over a few basic concepts of the thermodynamics of DNA flexibility and folding. And I thought about this quotation, which I had seen before. Thermodynamics is a funny subject. The first time you go through it, you don't understand it at all. The second time you go through it, you think you understand all of it except for a couple of small points. And then the third time you go through it, you realize you don't understand it, but it doesn't bother you anymore because you're so tired of the subject. So with that in mind, let me talk a little bit about the energetics of DNA bending. We tend to think about DNA bending as something that occurs on the scale of DNA lengths larger than a persistence length. And by the way, let me emphasize that in this talk, I'm going to be focusing on DNA molecules in the size range of roughly 100 to a few thousand, maybe up to 6,000 base pairs. So we're not talking about genomic scale DNA, we're not talking about chromatin. In principle, we can deal with issues having to do with DNA, sequence-dependent flexibility and shape, but I'm going to suppress that in this talk. So essentially, I'm talking about plain vanilla DNA that's between 100 and several thousand base pairs in size. So we tend to characterize DNA bending flexibility in terms of a quantity called the persistence length. Here, the symbol P. Elsewhere in the talk, I think it has the symbol L sub P. And essentially what the persistence length is a measure of a correlation length over which the directionality information in a polymer chain is lost. And so I think everyone here knows that this is about 45 to 50 nanometers for DNA at moderate salt, about 150 base pairs. These plots here are actually chains, DNA chains that were generated by Monte Carlo simulation, and you can see that even for chains that are a tenth of the persistence length, there's still a fair bit of bending flexibility. Once you get out to chains that are 150 base pairs or one persistence length, there's substantial flexibility. So these so-called bouquet plots, which Wilma Olson and others have published over the years, actually give, I think, a much more convincing or much more accurate picture of what the extent of DNA flexibility is than simply the persistence length alone. If you ask most molecular biologists what the persistence length means, they'll say it's the length scale over which DNA behaves as a rigid rod. Clearly, that's not true. So now 150 base pairs or 50 nanometers is pretty stiff for a polymer. So it's worth asking the question, what's the extent of compaction that actually takes place in, say, a vertebrate nucleus? And as we've heard, the nucleus has a diameter of about 5 or 6 microns, perhaps. But that nucleus has to contain 2 meters worth of vertebrate DNA. So this is really a 4 order of magnitude compaction problem, depending on how you want to calculate the amount of space occupied by the nucleosome-free uncoiled DNA. And of course, this doesn't occur spontaneously. This happens through the thermodynamic affinity of nucleosomes for the DNA, the protein-protein interactions that occur along chromatin, and so forth. But it's still a remarkable feat. Every time I tell undergraduates this, I always get this bug-eyed expression, right? Because it's equivalent to taking a piece of string that's maybe 0.05 millimeters in diameter and a length of 50 kilometers and compressing all of that into an object about the size of a basketball. And clearly, there's a lot of stored length in a chromatin fiber. These are actually AFM images of chromatin fibers that have been pulled to varying extents. We've heard this story several times this morning now. I'm not going to dwell on it, but I do want to focus on the fact that for the so-called equilibrium globule, the power law dependence of the so-called contact probability has exactly the same power law dependence that you get for cyclization of two free DNA ends when the DNA molecule is sufficiently long. And so you'll see that shortly, but it's still an interesting question of how you take some distribution of chromatin in a nucleus and even in simulations manage to get a structure that is truly equilibrated, unlike polymers in solution, where in fact you can reproduce both experimentally and computationally this minus 3/2 power law dependence. Bacteria have a slightly smaller hill to climb in terms of compaction of DNA. Here, though, that there's a role not only for proteins that stabilize bent DNA structures, such as the protein HU, and I think this will be the topic of Wilma's talk later in the week, but also through supercoiling of the DNA. You can see here in this image taken from one of Ruth Kavanaugh's electron micrographs, you can see sort of the outline of the E. coli cell, which has been lysed, and there are these large loops of DNA that extend from this area that was formerly occupied by the intact cell. So when you do the math here, it's about a 50 to 100-fold compaction problem, but the thermodynamics here are, in a sense, a bit more clear because of the fact that supercoiling itself tends to compact DNA. So this motivated a lot of the work that I'll talk about in the second half of the talk, where we were interested in trying to take complex nucleoprotein structures and see whether we could actually directly calculate the free energy cost of creating such a structure from simple models. So all of this is, of course, related to the problem of DNA looping. And we now know that DNA looping is possibly a ubiquitous mechanism for regulation in biology. And so there are lots of examples here of protein-DNA interactions that involve various sorts of loops. There's a well-known set of restriction endonucleases that operate via this sort of looping mechanism where two restriction sites have to be bound by the enzyme before it can actually do its enzymatic activity. This is an example of a repressor molecule bound to two very closely spaced DNA sites. There's only 50 base pairs of DNA here in between, but this is a very large complex. And so the complex forms more or less a straight line connection between the two sites. It's known now from lots of experiments, including those that were talked about earlier today, that there are loops that form in conjunction with transcriptional regulation in both prokaryotic and also eukaryotic cells. This is an example of some AFM images from my own group of Cre recombinase, a tyrosine site-specific recombinase, which I'll talk about further in a minute, bound to 2 target sites on a circular DNA molecule. Okay, so we've heard a lot about contact probabilities. I tend not to think so much about contact probabilities, but rather, because I'm a chemist by training, I tend to think about concentrations as a measure of thermodynamic drive. Okay, so what this graph shows are measurements of the Jacobson-Stockmeyer factor, which is effectively the concentration of one end of a linear DNA molecule in the vicinity of the other, as a function of DNA size. And I think this is possibly one of the experiments that Erez was alluding to earlier this morning. This is actually work done by Shore and Baldwin in the early 80s. And when they first published these experiments, they had a set of data points that sort of scattered around the graph. There really weren't any smooth curves to fit to the data because there weren't any theories available at the time that were comprehensive descriptions of the thermodynamics of cyclization. But they saw very clearly that these data points, and in fact they had data on shorter length scales, on the length scale of maybe 20 base pairs, that showed that this effective concentration, or J factor, actually oscillates up and down between the lower and upper dashed curves. The solid curve in the middle is the average over full helical turn, so it's basically a running average of this J factor. And so the explanation for this is very clear. If you have an integral number of helical turns, then it costs you relatively little or less free energy to bring the 2 ends of the molecule together and ligate them than if you have a half-integral number of helical turns, in which case you have to either overwind or underwind the double helix by half a turn in order to make the DNA helix continuous across the break. And so you have this oscillatory behavior for short length scales, but by the time you get out to about 1,000 base pairs, that oscillatory behavior starts to damp out. And in fact, what you expect is that the upper and lower dashed curve are going to approach some kind of asymptote, which in this case ought to be a linear dependence on a log-log scale. Moreover, the quantity J can be directly related to the free energy cost of cyclization, because J turns out to be nothing more than a normalized equilibrium constant for formation of this cyclized molecule. So if we have a free energy, there must of course be an energetic term, and there has to be an entropic term. And in the limit of short length scales, the dependence is dominated by the elastic part. In the limit of long length scales, the process is dictated by entropy, as the ends become more and more dilute from each other as the chain gets longer. And in fact, these upper and lower dashed curves approach an asymptote that's described by this -3/2 power of the length dependence that we've been talking about in connection with the equilibrium globule. Okay, so now the question is, how do you actually measure these J factors? Turns out that there are several different ways to do it, but the most rigorous methods use kinetics in order to get rate constants for the formation of a circle from a linear, and usually in a separate reaction, measure the rate constants for the formation of a bimolecularly joined DNA from 2 separate ends. So you can think of these red balls as being essentially activated ends. They could be cohesive termini, or they could be something else. And then J is gotten from this ratio here. So the question we asked is whether it's possible, in fact, to do this with a different enzyme system. And one of the motivations is to be able to do these kinds of experiments in living cells. Obviously, you don't want to have DNA molecules with free ends in living cells. You get all sorts of rearrangements. You get non-homologous end joining, perhaps, or other processes that you want to avoid. So we had been working with Cre and flip recombinases for quite a few years, and decided to see whether we could use Cre to interrogate the formation of DNA loops, in vitro at least. And perhaps that will lead to a technique that we can use in vivo if everything works out okay. Cre is one of a class of, a very large class, in fact, of site-specific recombinases called tyrosine recombinases. And that's because the protein relies on a catalytic tyrosine to carry out strand cleavage. This is the sequence of the recognition site or target site for Cre recombinase, a pair of 13 base pair inverted repeats that flank an 8 base pair asymmetric sequence. So if you have two copies of this LOXP site on a single DNA molecule in direct orientation, as specified by the the non-repetitive elements here, the asymmetric core, then when you take this linear piece of DNA, incubate with Cre, what you expect to get as products are a linear molecule and a circle. The circle corresponds to this piece of DNA in between, between B and C, which has now been effectively cyclized by the recombination reaction. And this is a crystal structure of a LOXP site complexed with a pair of Cre monomers. So there's a Cre monomer bound to each one of these symmetry elements in the active complex. This reaction proceeds via a 4-stranded DNA intermediate, which most of you probably know as a Holiday junction intermediate. And so this is a simplified cartoon here. It's not necessarily the case that the synaptic complex, this intermediate structure here, looks the way I've drawn. In fact, there's quite a bit of controversy associated with that, which I won't go into today. But in any case, you get initial formation of this 4-way DNA junction, which then is subsequently resolved in a second enzymatic step, effectively exchanging this red end for this purple end in the products. So we set about trying to measure the kinetics of this reaction under two sets of conditions. One where you have a pair of DNA duplexes that are synapsing and then forming products, and also a reaction where you have the two LOXP sites on a single DNA. So here the product is simply a swapping of a purple end for a black end, but here you actually get two topologically distinct products, a DNA circle and a linear molecule. And there are actually 8 rate constants in principle associated with the two reactions. So you have 2 rate constants for this step, 2 rate constants for that step, 2 for this one, 2 for this one. And we should be able to get J then as this quotient of rate constants. So we started out with the intermolecular reaction, and what we decided to do was to do this experiment using fluorescence resonance energy transfer. The reason for that is mainly that if you try to measure the kinetics of these experiments on gels, you can do it, but the noise level is pretty high because it's hard to get many data points. Also, there are other steps in the mechanism that I haven't told you about. And so in order to get the rate constants of interest, you actually have to fit the experimental data to a system of ODEs, ordinary differential equations. And the parameters for some of the other steps are already known from other experiments. So we're trying to measure 4 parameters, say, in an experiment where you already have 4 additional parameters that are fixed with a system of maybe 12 or 14 ODEs. Not an easy thing to do if you have a limited number of data points. So we needed lots of data, and the best way to do this was to do FRET experiments in real time, where we start out with donor and acceptor labeled duplexes, add Cre, and then look at the formation of a synaptic intermediate and then products as a function of time. And in fact, we can even extract some information about donor-acceptor distances, potentially, in the synaptic complex by measuring the apparent degree of quenching of the donor in this complex. And it turns out to be different in the donor than it is in the products. So we can actually measure independently in this experiment, or quasi-independently, the concentration of intermediate and product. So these are, in fact, fluorescence traces from experiments done with two different concentrations of substrate, 25 and 12.5 nanomolar. The data points are fitted to curves that come from the ODE solution. So overall, the fit is really quite satisfying. Now, for the intramolecular reaction, we took a different strategy for a number of reasons, and developed a new methodology which involves making covalently closed DNA with donor and acceptor labels. And so the way we do this is to flank the LOX-P sites with a pair of restriction enzyme recognition sites for the enzyme BBVC1 and BSM1. And there are mutant forms of those enzymes that will nick only one DNA strand. So by flanking the LOX-P sites and then nicking with these mutant forms of BSM1 and BBVC1, we generate vicinal nicks and single strands that then can be displaced by appropriately labeled, fluorophore-labeled oligonucleotides. And then we seal everything up with T4 DNA ligase. And I'm probably the only person on my floor who still uses ethidium bromide cesium chloride gradients. But it turns out that that's the best way to purify these plasmids because you can get rid of all the junk. And so what you end up with are plasmid DNAs that are labeled site-specifically at two sites. So now we can do the same reaction. This could be done on circular DNA, it could be done on supercoiled DNA, but what I'm going to show you here is just linear DNA. We linearize the plasmid at a specific site. As before, we measure the time-dependent Fret signal, which contains a contribution from the intermediate and also from the product. And this is what the curves look like for two different sizes of loops. So in one case, the curvilinear distance between the sites is 3 kb. In another case, the curvilinear distance is 870 base pairs. These are the rate constants. What I'd like you to notice is that the forward rate constant for this step, going from the free DNA to the looped intermediate, the rate constant for that forward step is very dependent on loop size. However, the rate constant that takes you out of this intermediate state is essentially independent of loop size. So this is where you can actually pick up the effect of a DNA loop on the rate of the reaction. So now, putting all of this information together, we can estimate what the Jacobson-Stockmeyer factor is for the different size loops. This is the 870, this is the 3 kb loop. And we can also ask how those values compare with theory. Well, it turns out there aren't good theories for looping in this size range, at least not yet. But we can compare at least with values of the J factor for cyclization. In fact, since we're dealing with relatively large loops here, and the capture radius for the enzyme is probably small compared with the size of the loop, that's not an unreasonable comparison. And so we were very, very pleased to see that the experimental values agree really quite well within error with the theory for J as a function of DNA length. at least for cyclization. We don't expect huge changes in these values when we consider looping versus cyclization. And by the way, these values come from a procedure that John Marco worked out, which is essentially a convergent sum that you get from a transfer matrix calculation. Okay, so now we felt the next thing we decided to do is to try and address the theory. Because if we go back to these J-factor calculations, it's pretty clear that this J-factor could be affected dramatically by the topological state of the DNA. So if the DNA is supercoiled, or if you're carrying out the reaction, say, in a special case like a knotted DNA molecule, that could dramatically affect the J-factor. And there's no theory that takes us there. So some years ago, I worked on a theory with Yonglei Zhang, who was then a graduate student at Yale. And we worked out a methodology for calculating the free energies of looping for small DNA loops. And so the equations are here, but basically what it comes down to is you have a free energy of loop formation, which involves the energy of the energy minimized loop. So you take a loop of some arbitrary conformation. The conformation is going to be dictated in large part, because these are small loops, by the structure of the protein that's mediating the loop. You take a model for that structure, you energy minimize it, you get a value of the energy at the minimum, and then the entropic part of this free energy comes from an expression that involves the determinant of some matrices. And those matrices contain things like force constants for bending, force constants for twisting, all of which are part of this Hamiltonian. So there are various factors and terms that appear in this Hamiltonian for the DNA that go into the calculation of the A and B matrices. And the advantage of this approach is that it's an incredibly fast way to calculate J. So it's about 4 orders of magnitude more efficient than calculating the same quantity by Monte Carlo simulation. The reason for this mainly is if you want to calculate probabilities of loop formation in a Monte Carlo simulation, you have to deal with the problem of sample attrition, which is very high. You lose a lot of configuration simply due to the fact that they don't loop or they don't cyclize. John?
That comparison must be the particular length scale.
Well, yes. So these calculations actually it does scale with length, but it's so fast that we don't care.
Comparison between the semi-classical and the Monte Carlo must switch with the length of the DNA, no?
Yes, that is true. That is true.
Yeah.
That is true, and it will depend on what the shape, where you are on the J curve, basically. So that is correct. In any case, this is a calculation that can be done essentially in wall clock seconds, whereas the Monte Carlo simulations take hours to days, depending on the thermodynamic cost of forming the loop. So what we had hoped was that we might be able to extend this to a regime where the loops are sufficiently long to incorporate significant amounts of supercoiling or other topological constraints. This is basically a slide that shows the application of the harmonic approximation theory, this theory, we call this the harmonic approximation, to some in vivo experimental data. The bottom line is that we were able to extract from fitting various experiments the effective persistence length of the DNA in living bacterial cells. And it turns out that there's about a 40% reduction in the effective persistence length of the DNA that the theory can account for by fitting the experimental data. Okay, so this harmonic approximation, although it's fast and admits a base pair level description of the problem, has some significant limitations. Essentially, once you get out to about 300 base pairs, these systems don't behave harmonically anymore. And so the notion of using a harmonic approximation breaks down. Also, there's no excluded volume. So if you want to include supercoiling, you can't do that. And so we had to go back to computer simulation to address these longer loops. And of course, now we have to consider the possibility that there will be topological equilibrium as well. If you randomly close a DNA chain, you expect to get some distribution of supercoiling, but you may also, if the chain is sufficiently long, get some knots or some links if the concentration is sufficiently high. And in fact, you can even observe these in experiments. These are Cree recombination experiments where we started out with a relaxed DNA circle, and then the recombination products include not just circles containing an inverted sequence, but also trefoil knots, 5-noded torus knots, and so forth. And so you can see here that the vast majority of the product DNA is unknotted, but we'd like to be able to account for these relatively small proportions of various knot types. And this just shows that you can identify these as knots by cutting the DNA out of the gel and then coating the DNA with Rec A and visualizing in the electron microscope. Okay, so we set ourselves two objectives with the theory. The first was to see if we could reproduce the minus 3/2 power law dependence for the cyclization probability for long chains, and then to look and see whether we could also accurately determine the knotting probabilities of relaxed plasmid-sized DNAs for different knot types. Okay, so that reminded me of a second quote I wanted to include in the talk, which is that nothing in life is certain except death, taxes, and the second law of thermodynamics. All three are processes in which a useful or accessible form is transformed into useless, inaccessible forms of the same quantity. That's not to say that these processes don't have fringe benefits. Taxes pay for roads and schools, unless you're a Republican. The second law of thermodynamics drives cars, computers, and metabolism. And death opens up tenured faculty positions. So like everything else in life, the problem for us is not calculating the energy of loop formation or the energy of knotting, but rather the entropy that's associated with that process. So we use a coarse-grained description of a DNA polymer. This is about 60 base pairs of DNA, which we model as two rigid segments connected by a semi-flexible joint. This model has about 7,200 degrees of freedom. This has about 2 degrees of freedom. And for a 3 kb circle, you have about 200 degrees of freedom overall. So that makes the problem much easier. The basic key to the approach is to use a thermodynamic cycle argument. So what we do is we use a technique called thermodynamic integration to get the difference in free energy between 2 points along either the blue curve or the black curve. So the black curve might correspond, for example, to an unknot. The blue curve might correspond to, say, a trefoil knot. So that gives you the free energy difference between, say, points B prime and B or A prime and A. The problem is to get the leg of the thermodynamic cycle that connects A prime and B prime. And so this thermodynamic integration is controlled by a perturbation parameter lambda, which you can think of as being essentially the inverse of the absolute temperature. It's not precisely equivalent to that, but it's analogous to the temperature in the simulation. So at finite temperatures, we integrate along these curves, but at low temperature, we have to get the free energy difference between A prime and B prime. So how do we do that? Well, to do that, we use so-called normal mode analysis. You calculate the normal modes or eigenmodes of each system at a low temperature, And for a system that behaves harmonically, you can rigorously calculate the partition functions of those two states. And so once you have the partition function is essentially exponentially related to the free energy, then you have the free energies. So if you have this leg of the cycle, this leg of the cycle, and this leg of the cycle, you can get the fourth leg by difference at finite temperature. And that's what we do. The thermodynamic integration is applied to a model of a circular DNA as implemented in a Monte Carlo approach that I worked on with Alex Vologodsky many years ago. Essentially what we do is we fold up in random ways you don't see the individual segments here, so there are about 300 segments in this chain. And what we do is we fold the polymer along cords that connect vertices of the circular chain. So that's one fold, there's another fold, and you keep doing this, and after some period of equilibration, the system just fluctuates. So what you're seeing here in real time is the folding of the polymer into an equilibrium ensemble of conformations starting from a planar polygon as the initial configuration. And I left out the Bob Marley, which some people find disturbing. Okay, and then the rest is handled by numerical integration of this expression. This is the partial derivative of the potential with respect to the perturbation parameter, integrated between two states, A and B, two values of lambda. So we can go from low temperature to high temperature, or we could do the reverse. It doesn't matter which direction we go. The difficulty of, the reason why we can't use thermodynamic integration alone without normal mode analysis is simply because you can't cross topological barriers with thermodynamic integration. You can't handle phase transitions and so forth. So we use the normal mode calculation to get us from the unknot, say, to the trefoil. And this just shows, this is one of Stefan's movies, which shows how the chain behaves as a function of the perturbation parameter. Remember, we're effectively going down in temperature. And what you can see is that there's a freezing out of the modes in the chain. And then by the time we get to lambda values of around 50 or so, the system behaves harmonically, which is something that we can actually verify numerically.
Is that a phantom chain, or there's repulsion?
No, there are interactions that
Repulsion,
yeah, that keep the chain from violating excluded volume. And you can use essentially any model you like for long-range interactions. Here are just the lowest frequency normal modes for the trefoil and for the figure 8 knot. And so the eigenvalues or eigenfrequencies we get from diagonalization of a Hessian matrix, which is basically the matrix of second derivatives of the potential energy. So how well does all of this work? Well, I think Stefan had some concerns when I was putting the talk together that people in the audience might find the data too good to believe. We were actually astonished ourselves. The smooth curves in this plot are actually the free energy, and entropy terms of cyclization as a function of chain length. So this goes out to about 6,000 base pairs. And these values, the smooth curve values, come from the transfer matrix approach I talked about earlier. So with a sufficiently large number of turns, you can make the transfer matrix essentially exact. And the data points are the values that come from the normal mode thermodynamic integration method. So they're essentially indistinguishable. I mean, there's a little bit of noise, but it's on the order of something like 0.01 kT. Even more interesting, I think, is the fact that we can directly calculate the free energy cost of forming a knot. So these are results for a 3 kb DNA circle. And what we're comparing here are the free energy of knotting values that we calculate using the pneumatic method, which is our acronym for this new procedure, and comparing those values with the knot free energies that you would get from the frequencies of knots appearing in a simulation where you allow free segment passage. We call this RSP or ESP, equilibrium segment passage. There are no strands in the model, just segments. And again, so in a sense, the equilibrium segment passage model has been used before by Alex Vologodsky's group and also by Andre to calculate free energies of knotting. In a sense, we've used that as our gold standard here. But the advantage of the pneumatic calculations is we can start with any arbitrary topology, and we can remain within that topological space throughout the entire simulation. We don't have to account for what might happen to delta LK, for example, as two segments pass each other. So we can do this for loops, we can do this for knots, we can do this for essentially anything we want, and that was the goal. So I'm probably out of time. Let me just quickly summarize that we now think we have a good method for measuring the free energy of DNA loop formation, potentially one that can be applied in living cells, and that's to use Cree site-specific recombination. We need methods for computing the free energies of loop formation and other topological states as a complement to those experiments. And we're very pleased that we think we have such a method. There are actually a set of methods that can be used here. Even though the pneumatic method would work for any size DNA, probably it's still more computationally efficient to use the harmonic approximation, so we're not planning to abandon that. But the pneumatic approach is valuable because it allows us to get into a regime of chain length which is very difficult to handle by any other method. So I think I'm going to end there, and thank you for your attention. This is a picture of my daughter modeling the latest in sleepwear from notplot.com. So anyway, thanks.
Steve, thank you very much. Yes, Aris, please.
I thought you might find the following interesting. So actually, I was really interested in these loop factors like 4 years ago when we first got the IC data, because it turns out you get lots of these fragments, and actually the kind of the most common thing to happen with these fragments is that they form a little loop.
Right.
And so you can ask yourself the question essentially is how often do they form this loop versus the straight, how often do you essentially see the straight form versus the loop form as a function of the fragment's length? And at the time, I observed that it seemed to tail off with a scaling of -3/2, which seemed right. But then I never kind of reported it. But it seems like actually something that could be a potential alternative source of data of this type.
Right. So that's exactly what one would expect, I think. One of the things that we've noticed in these calculations is that the apparent J value that you would get out of the simulation isn't terribly sensitive to the capture radius. As long as what you're doing is calculating the concentration of one site in the vicinity of the other. It doesn't matter whether you choose a sphere that has radius 10 nanometers or one that has radius 30 nanometers. and I'm expecting, we haven't done a thorough analysis of this yet, but I'm expecting that you ought to get pretty much the same -3/2 power of the length dependence as long as the DNA is relaxed. And in your case, it would be. One of the ultimate objectives of this is to see whether if you have, for example, some protein constraint, and now you introduce a site-specific recombinase, you can form potentially a fairly complex structure and ask what's the J factor going to be for the recombinase given those other topological constraints. And so that's essentially where we're heading. But I would expect that we ought to be able to fit the data you're talking about pretty well.
Based on the Cree experiments that you guys have done, what is your sense of the persistence length of in vivo chromatin?
We haven't done the experiment with a chromatin substrate. So that's something we would like to do. But so far, everything we've done has been in vitro. Yeah, Cree may not necessarily be the best system to use, by the way. It may not be a universal tool. What I think we would like to see are other enzyme systems developed for this sort of probing. But Cree is a lousy enzyme. It's really slow. And so we got lucky that we were actually able to measure kinetics on this particular time scale using manual mixing. And in fact, you could probably see in the 870 base pair data, the curve drops very sharply. So we can't measure the initial transient, but we can measure the plateau pretty reliably. But if you're interested in dynamics on a more rapid time scale, it may be better to use a different sort of probe. And so hopefully this will stimulate other groups to develop similar approaches using, for example, type 2 restriction endonucleases or something like that. John?
So we're in a maths institute, so for once I don't have to apologize for asking this question. But when you talk about concentration in
Yes.
these experiments, is it a concentration in three-dimensional XYZ that's important?
That's exactly right.
So it's not a concentration in XY, the six-dimensional XYZ and three orientations.
Oh, I see.
level it
I
must
see,
be, right?
I see. Okay, so for systems of this size, we can ignore the
Which
torque.
size?
So in the say 1 to 6 kb range,
Uh-huh.
we don't worry about the torsional.
So you think it's
Contribution.
gone?
Yeah, it's effectively gone because there's essentially not much energetic penalty associated with over or underwinding the DNA.
So when's the switch over then? Because I mean, sure, Baldwin, exactly the phasing's the twist, right?
Right.
When do you have to switch the dimension
Well,
of your density?
so yeah, it's probably not so well defined. You know, I think probably. You would have to ask me a question about something at the beginning of the talk. You know, it's probably right around here where you get the convergence of the maxima and minima. So what is that DNA length? It's close to 1,000 base pairs. And that's the regime I focused on today for this talk. But for DNA loops in this size range here, we would clearly be better off using the harmonic approximation because then we wouldn't have to worry so much about how we're going to do thermodynamic integration over this additional torsional degree of freedom.
Okay, so we leave more questions for the break, I think, and then so we have a coffee break or tea, afternoon tea. It's also the poster session. So if you have your tea, visit posters, discuss posters, and discuss between yourself. Thank you again, Steve.